Hello, my name is Mr. Chipman, and this is AP Bio Review Unit 6, Gene Expression and Gene Regulation. We have a test tomorrow. Hopefully you are prepared. Hopefully these videos have helped you. If they have, please like down below. Also subscribe to my channel where I'm always posting new biology content. Give me a comment down below. Tell me how helpful it was. Those are encouraging to me as a teacher. So I'm really thankful that you've been able to use these videos for your review, whether you're my student or you're someone else's student. Let's talk about this stuff. DNA is a molecule that stores genetic information. It's made up of 3 parts. Well, each DNA is made up of lots of parts called nucleotides. Those nucleotides are made up of sugar. That sugar is called deoxyribose. It's a 5-carbon sugar, a phosphate, which is just a phosphate, and nitrogenous bases, A's, T's, C's, and G's. A and T are adenine and thymine paired together. G and C, guanine, cytosine, paired together. Thymine, cytosine are called pyrimidines. Adenine, guanine are called purines. Moving on a little bit more structure of DNA. This shows you those pairings. There's hydrogen bonds formed between the 2 strands of DNA. DNA is double-stranded, and it's double helix in shape, meaning it coils up, but it's better to see it flat like this. C and G have hydrogen bonds between them, so do A and T. These hydrogen bonds are pretty easy to break, but they like each other, and so that's necessary for DNA replication. But the bonds between the sugar and the phosphate are super strong, and so that is important. Those are covalent bonds, meaning that they share the electrons. So let's continue on. One thing to note too, notice that these 2 strands of DNA go in opposite directions. We're going to talk about that directionality in just a moment as we talk about DNA replication. But they're anti-parallel. They line up parallel to each other, but they're going in opposite directions. Let's continue on. A couple things to note about eukaryotic and prokaryotic DNA. Prokaryotic DNA, the bacterial chromosome is a single circular chromosome, whereas that's in prokaryotes, and they have this plasmid DNA, which kind of is like extra genes, whereas in eukaryotic cell, all the DNA is found in the nucleus, and there's multiple linear chromosomes there. DNA replication process that makes a copy of the DNA. Why would you do that? Because you want a copy of cell. Of course, you would need 2 copies of DNA to make 2 cells. This is mitosis. Remember that process? Well, the DNA replication in eukaryotic cells is called semi-conservative, meaning that you save half, conservative, save half, semi. So here's the old strand. That strand is going to divide in 2 and serve as each half is going to serve as a template of the new. And so you have strand here, strand down here, and you have 2 identical strands when it's over. Both of those contain all of the DNA, or at least theoretically. There can be problems with that replication. We'll talk about that when we get to mutations. Moving on, let's talk about the directionality of DNA replication. DNA is always read from 3 to 5, which is important. It's always read from 3 to 5. The enzymes are going to sit down on top of one of those strands and read it from a 3 and go to the 5. It's going to add new bases. The new strand is going to be made from 5 to 3. So notice the difference there. I'm sitting down on 3 and I'm moving to 5 if I'm an enzyme, but I'm adding new bases 5 to 3. Why are we doing it opposite? Because they're antiparallel. Don't forget that. DNA is going to be unzipped, but it's only being unzipped in a particular direction. Notice that one of these strands is being added in the same direction that it's being unzipped. And so there doesn't have to be any kind of stopping there. You can think of this as it just continuously goes. Sometimes you'll see this called the continuous strand or the leading strand. Leading strand is a little less helpful, I think, but it is the term that's been used a lot. I like continuous much better because you can think of it as since it's going the same direction as DNA is being unzipped, it doesn't need to stop going. However, the other strand is also read from 3 to 5 and is being added 5 to 3, but it's the opposite of the leading strand. And so basically as it unzips a little bit and adds some new DNA, unzips a little bit more, adds some new DNA. And so it's a discontinuous strand. It can only go backwards. It's going the opposite direction that DNA is being unzipped. And so as new sections are unzipped, it's going to lay down more new strands. Sometimes you'll see these individual fragments, these little sections that are laid down called Okazaki fragments. Haven't seen that in the new DNA literature, though, but it is important to understand that one of those strands is happening continuously, the other is discontinuously. Sometimes you'll see that discontinuous strand called the lagging strand. Again, it's not as helpful, but it is the language that you'll see a lot. And so go ahead and know those terms, but understand it in a continuous, discontinuous idea. Again, here's the directionality. You have this DNA polymerase that's adding strands or adding new bases from 5 to 3, right? But it's reading this strand, right? It's reading this bottom purpley strand. It's reading it in order to add new bases. So it's reading TCAG, but it's adding from 5 to 3, AG. Okay, I think you understand that. Let's talk about a few of the enzymes that are involved in DNA replication. Topoisomerase is an enzyme that basically flattens the DNA out, relaxes the DNA. These single-stranded binding proteins are what they're going to do is they're going to allow for the DNA to temporarily not bind to itself again. It's going to keep it open so that the new strand can be made. Helicase is the zipper. It's this little triangle right here that looks like it's making a wedge between the two strands. That's because it is. It's unzipping the two strands of DNA. DNA polymerase is adding more nucleotides. You could think of it as creating the polymerase, right? What else? DNA primase. It adds an RNA primer. Why would it do that? Well, the primer is where you start, right? So it's priming the DNA, getting it ready. On the leading strand or the continuous strand, this only has to happen once. But on the lagging strand, each time a new fragment is made, DNA primer has to put down a primase, or primase has to put down an RNA primer in order to get that started every time. And then ligase has the prefix Lig, which means to bind like a ligament. Think of a ligament binding something. That ligase is going to come and bind those fragments together, those fragments on the lagging strand. Moving on. Let's talk about the flow of genetic information. We've talked about DNA making a copy of itself. I love this picture, by the way. It's fantastic. It helps me to think about things. Some of this stuff is weird, but for the most part, it's really good. DNA makes a copy of itself. That's called replication. What if DNA makes RNA? Well, that's called transcription. It's the same language. Imagine if someone was sitting right next to me, writing down every word I was saying. They're transcribing what I'm saying. It's going from voice to print. It's a different form, but it's the same language, right? They're transcribing it. DNA and RNA are the same language, both nucleic acids. It's just a different form. But when you translate something, you're going to a different language. And so here we have RNA going from RNA to a protein. That's a completely different language, right? We have nucleic acids to protein, different language. We're going to talk about transcription and translation. Sometimes you'll see these two together called the central dogma. It's not very helpful. Transcription, translation, here they are. Again, DNA transcription and translation together are called protein synthesis. The purpose is to make a protein. How do you do that? Well, you go from DNA to RNA, that's transcription. And you go from RNA to polypeptide, which is translation. So yeah, some of this stuff that you'll see in the literature was really confusing and not helpful at all. When you transcribe, only one of the strands of DNA is being read. Because remember, DNA is double-stranded, but RNA is single-stranded. And so only one of those is being serving as a template. It's called the template strand. The other strand is called the coding strand. It seems like you're doing the same thing. And so one of the things that my students and I came up with, and actually it was their idea, and I thought it was great, it's kind of like, it looks like the code, right? But it's not exactly. Why is it not exactly? Because RNA has U's in it instead of T's. But otherwise, it's the same thing. Just put a T where there would be a U. So it's kind of like a code. I don't know if that works. I don't think there's going to be a whole lot about that. You're definitely not going to give a multiple choice. What is this strand called? Template or coding? That's not the AP way. Probably be in the literature, and you'd have to like discern. But as long as you understand the difference between transcription and translation, I think you're going to be safe there. Transcription or translation, again, reads that RNA, converts it into polypeptide. We'll talk about how it does that. Transcription, big enzyme that involved in that is called RNA polymerase. What is it doing? It's making a new polymer. What is that polymer? It's RNA. And it's only reading a section of the DNA. That's important. It's not reading all the DNA, it's just reading a section. That section is called a gene. Gene codes for a particular trait. How do you get that trait? Well, protein. So it's going to make a protein. That's transcription. Moving on. Here's transcription again. Pretty much showing the same thing. Notice bases are added from 5 to 3 again, read from 3 to 5, same as DNA replication. There are different types of RNA. There's messenger RNA. Messenger RNA is a message. It is the message that DNA has for the cell. It's about how to make a protein. It's like the intermediate. It is what is being made in transcription. This is the mRNA. rRNA, ribosomal RNA, it's part of the ribosome. That's all you need to know about it. Transfer RNA, what does it do? It transfers the amino acid to the ribosome during translation. So it has a job. It's called transferring. It's a special piece of RNA. Almost has an enzymatic function. We'll talk about that later. Let's talk about prokaryotic transcription. Prokaryotic transcription, basically the idea here is that you understand it's the same idea. You're still making an RNA from a DNA, but that RNA can be read immediately by the ribosome. Whereas in eukaryotic transcription, there are several things that have to happen to that transcript before it can be made into a protein. We're going to talk about those several things. What are they? Well, here they are. There's 2 of them. First thing that's going to happen is a poly A tail is going to be added to it. What does poly A mean? Literally means lots of adenines, 50 to 250 adenine nucleotides added to the 3' end. There it is. All this other stuff, I don't even know what it is. Well, I've learned it at one point, but you don't need to know it. Poly A tail, and the other end has what's called a GTP cap, or you could just say a 5' cap works as well. But GTP cap, just so you don't get confused when you see that, GTP is just guanine triphosphate. It's just kind of like the way it was described to me when I was learning all this stuff for the first time, it's almost like a reverse backwards section that's added to that section of RNA. Don't worry about that. The purpose of the poly A tail and the 5' cap is to protect the message. The 5' cap is also going to signal the ribosome, hey, it's time to do this. The poly A tail, I like to think of it as like Pac-Man bits. It's like if this is the protein coding sequence, I want to protect that, and the enzymes in the cytoplasm are dangerous. They're like Pac-Man. They're trying to eat up the letters, and the poly A tail is going to give them something to chew on. Think of it that way. And then lastly, what is happening to these pre-messenger RNA? The introns are taken out and the exons are left in. The introns are these sections of DNA that are non-coding. They don't code for anything, at least we know of. And the exons are the important part. You can think of it as a 500-page book that has 80 pages of text and 420 blank pages. The introns are the blank pages. Take them all out. They're unnecessary. Combine the other 80 pages, make you a nice book with a 5' cap and a poly A tail. You got a mature messenger RNA ready to do the work. Actually, it's not going to do the work. It's just going to be read like a book. It's going to be read by a ribosome. And this is alternative splicing. What is this? This is the idea that the exons, the coding portions, can just be mixed up to make different genes. So you have, or to make different proteins. So you have one gene that could code for multiple proteins. Sometimes you'll see these like protein families. That's about it. There's no more depth to go into on that. You may see a question that would like mention this and then talk, the question may be like, well, why are there different proteins for one gene? And you would think, oh, okay, well, maybe there's alternative splicing involved in that. That would be probably the extent of it. Translation. This is where you take the messenger RNA and you create a protein. How do you do that? Well, the messenger RNA is arranged in groups of 3. These groups of 3 are called codons. And these 3 things are nucleotides, A's, U's, C's, and G's. And each codon, each group of 3, codes for a particular amino acid. How do you know? Well, let's go find it. It's on here somewhere. No, it's on here somewhere. There it is. Wow. I went through a lot of things to get to the codon chart, but here it is. This is all the codons. Some guy discovered all this stuff. I forgot his name. I think his name's Marshall Nierenberg, actually. But he discovered the codon chart. Basically, there are 4 letters, and there are groups of 3, so there are 64 different 4 to the 3rd different codons one of those is a start codon. Start codon also codes for a particular amino acid. Just note that's always AUG. And then 3 of these codons are stop. They tell it to stop. And the rest of the codons code for amino acids. You don't have to memorize this. You'll be provided with it if it's necessary. But let's just look like C, C, C would be proline, right? Okay, that's it on that. Let's go back up to the things. Steps of translation. There are 3 steps to translation: initiation, elongation, and termination. And they do exactly what you think they do. This is initiation. Actually, this picture is just not helpful. Let's talk about translation with prokaryotes real quick. The big thing is that it's happening at the same time that transcription's happening. It can't with eukaryotes because with eukaryotes there has to be some stuff added, right? Poly A tail, 5' cap, and introns are taken out, exons are to get put together, and that happens in the nucleus, and that mRNA has to go outside of the nucleus in order to be translated. In the bacteria, all that's happening in the same place. That's the big difference. Retroviruses. Well, let's talk about retroviruses real quick because retroviruses are viruses that are RNA viruses and they infect eukaryotic organisms. How do they do that? That are DNA based. Well, they carry with them the code for this little protein called reverse transcriptase. Transcriptase does transcription. Reverse transcription is RNA to DNA, and that DNA is encoded for, codes for a protein. That protein is made and it makes more viruses. We're not going to talk about the viral life cycle right now, but just so you know, general idea, retroviruses are RNA viruses. They're particularly dangerous because RNA is not very stable. It mutates really quickly. Let's go back to the steps of translation. Talk about initiation. Initiation is when the 5' cap binds to the lower subunit of the ribosome. And there's all this other stuff with EPA, all that's gone from the literature, thankfully. But what this blue blobs represent is the ribosome. And notice here's the 5' cap, and AUG is going to be the first codon. And this little thing right here, or this little bar right here, represents the tRNA. tRNA, the transfer RNA, bringing the amino acid into the ribosome in order for translation to occur. So AUG codes for methionine. The next codon is going to be something else, but initiation is the beginning of this process, and it looks just like this. It's just going to set it up, get it ready to go. And there's the codon chart. There's tRNAs, just talking about those a little bit. tRNA, there are 61 different tRNAs. Where'd I get 61 from? Well, it's this minus 3, because there's tRNAs don't carry around stop signs. They just carry around amino acids. And so there literally is ACC tRNA. What is it looking for? UGG. This is the codon. This is the anticodon here. It's the opposite of the codon. And it only has one thing. It sees UGG, UGG. It carries around tryptophan. There's tryptophan right there. That's the only thing it does. It's going to go get some more tryptophan and just wait until it sees UGG again, which is probably right there, actually. So there it is. So yeah, it's just going to wait till it sees another one. That's all it does. And they're just going to keep bringing in new amino acids, going to add to this growing chain. That you could say that chain is growing longer. That's called elongation. That's it. That's all elongation is. It's just a growing polypeptide chain until it gets to the stop codon. What do you think that's called? Termination. That's it. There's this little release factor that's going to come down. It's going to bind with the stop codon. The polypeptide's going to break off, and it's going to fold up and do the polypeptide things. That's it on that. Regulatory sequences. There are some things that regulate transcription and translation. We're going to talk about a few of these things real quick because I know this video is getting pretty long. One of those things is called an operon. Big thing with an operon, it happens in prokaryotes first, and the idea behind an operon is to regulate the expression of a gene. Gene expression is the creation of a protein. Usually an operon has some sort of piece to it that is going to bind to this repressor, and the repressor is going to keep that process or keep that gene from being transcribed, right? This like Z, like Y, like A are these genes. And when that thing when this molecule binds to the repressor, it's going to cause the repressor to lift off, allow RNA polymerase to do its job. That is the real short version of an operon. The big idea behind the operon, you're not going to be shown a lac operon more than likely. You're going to be shown something else. And so, big idea behind the operon is just gene express or control of gene expression or gene regulation is another way to say that. So as long as you understand that what you're looking at is there to control the expression of that gene, then you can kind of piece out what the picture's showing you, right? And words like operator and repressor should be just common words as long as we don't overthink things. So there's another operon. You can kind of see the lac operon controls the expression of genes that break down lactose. Well, obviously, if there's no lactose, then those genes don't need to be expressed. When lactose is present, the repressor is going to lift off. The RNA polymerase is going to transcribe those genes. They'll be translated into proteins that break down lactose. When lactose is gone, the repressor is going to bind back, and it's over. That's an operon. Only found in prokaryotes. In eukaryotes, we also have control of gene expression and our gene regulation. One of the ways you see this is in cell differentiation. Not all of our cells are the same. How are they different? Why is a skeletal muscle cell different than a thyroid cell? It's because not all of the genes are being expressed. Thyroid genes are not being expressed in skeletal muscle cells. And so there's a control, right? The genes that are uncontrolled are called pluripotent cells or stem cells. These are cells that are able to express all the genes, but eventually there's going to be a high level of control that is exacted on those cells just to create efficiency, right? When you walk into a giant library with 50 billion books, but you only really need to be reading 6 of them, you're going to keep those 6 off the shelf and you're going to keep them close as opposed to going and finding them every time you needed them. You're just going to shut down the rest of the books, and that's kind of what's going on. We're going to move on. And so they're basically transcription factors. You can transcription factors are things that cause transcription to happen, and so anything that blocks those transcription factors are going to be some sort of gene regulation. Repressor blocks transcription factors. Just as long as you understand the language, the thing doesn't have to be scary, right? And so these small RNA molecules, sometimes you'll see them. Like miRNA is just microRNA. Small nuclear RNA is another one. These are just genes, or these are little sections of RNA that control transcription and translation. That's it. Don't get too crazy on it. Moving on. Mutations can obviously control or can cause differences in transcription and translation. And so, a normal protein is going to behave a certain way. A mutated protein can't, because why not? Well, DNA mutation is going to cause a different RNA, which is going to cause a different protein, which is going to have a different shape, which is going to have a different function. It all goes down to what is the shape of this molecule? How do I know its shape? Because it has a particular chemical makeup. How does its chemical makeup decided the DNA? And so it all traces back to the DNA. There are types of mutations. What are those types of mutations? Point mutations, change in a single base. A silent mutation is a change in a single base that does not change the amino acid. So notice AAG and AAA code for lysine. So that's not a problem. It's called silent. ATC is UAG. So instead of TTC, if it was ATC, that would code for a stop codon. Be a nonsense mutation, it's an early stop codon. And a missense mutation is a mutation that causes a change in a single amino acid. Not concerned that you know the difference between conservative and non-conservative. And there's a frame shift mutation. This is where you have an insertion or deletion of a single base or a section of bases that causes everything to shift down, causing that section of DNA to be read differently. This is on purposely showing you a lot of histidines together, but if you take a single A out, or in this case you insert an A, everything shifts over, causes all those to be read differently, which is not good for the DNA. Polyploidy is where you have more than 2 chromosomes in a pair. With eukaryotic organisms, this is bad. We're going to talk about that. But with plants, it sometimes can be good. With plants, polyploidy is not necessarily a bad thing. Sometimes it can lead to sterility, but for humans, we like that sterility because the banana doesn't make seeds. If you've ever eaten a banana with seeds, it's kind of gross. So a banana with seeds or without seeds is much better. So we make a sterile banana, and it's better for us. Um, Three types of horizontal gene transfer. This is a long video. I understand that. So there's transduction. Transduction is this is all three of these are prokaryotic, by the way. Transduction is when a virus introduces DNA into the new cell. Conjugation is when two cells exchange DNA, and transformation is when a cell takes up DNA from the outside. Did this with the bacterial transformation lab where we made glow in the dark bacteria. Not very many, but we made some. We're going to just skip past all these things. Translocation is where a section of a chromosome relocates to another chromosome. Probably not going to see this on the exam. I've never seen it. This will be the year, I'm sure. But just you can look at the word and understand what that means. And we're going to skip past this, but this is just the idea that because of the way that viruses infect cells, if a person is infected with multiple viruses, that can cause those viruses to combine and like mutate into things that other animals can catch. And this is, talking about the different kinds of flu that are out there. It's not going to be anything complicated at all. And then the last section of the unit was biotech, and biotech had to do with just different technologies associated with DNA. Genetic engineering is the change of DNA in an organism. Gel electrophoresis is one of the labs that we did in class where we drug the DNA through this gel. DNA has a strong negative charge, and so if you put a positive charge at the other end, it will drag through this gel, and the large pieces won't be able to make it as far as the short pieces. And you'll have something that looks like this. There are these enzymes that cut the DNA up into fragments. Every time it sees a certain section of DNA, it cuts it, and those fragments can get measured using gel electrophoresis. Polymerase chain reaction is literally polymerase, right, making new DNA. Chain reaction is a chain reaction. It's taking a section of DNA and copying it. PCR is a technology that was invented in the 80s in order to make lots of copies of a particular gene. We did not do this lab, but some groups do a PCR lab. And bacterial transformation, there's that glow in the dark bacteria. We did that in class. Hopefully, this has been helpful. I know that it was a little rushed, particularly there at the end. If you have questions, please comment down below. Good luck on your test tomorrow. Hopefully, it goes well for you. Thanks for watching.